I'm just about to fail the first test because this was switched off so that I could go outside. Have I managed to switch it on? Thank goodness for that. I've been pressing buttons all the time that Deborah was talking, just in case I wasn't getting it right. So this is my reaugural. And it's a pleasure to be reaugering with you. So what I'd like to do this evening is to give you a little bit of a personal tour of microbiology. And when I was asked for a title, I reflected for a little bit, and then I thought, let's come up with something a little jaunty. So, you know, what's bugging you? And then from bags to bones with towers in between, that sounds okay. And then I came to put it together. And I thought, why did I provide a title like that? There were so many complications in that title that made the talk one that I had to assemble from scratch rather than just draw off my previous inaugural shelf. So this truly is a reaugural. So what is bugging you? It was really good when I did what everyone does and scanned the internet to Google and found a really good example here that connects my talk directly to an exhibition in Brighton. An exhibition from the School of Artists called the Prokaryote, the Prokaryote movement. And in this movement, they use microbiology to create images. And these are really big agar plates, microbiology plates. And the artist has lain on them and then incubated them, and then I trust disinfected them, illuminated them with an appropriate backing light, and did many other things. But what's great is it really illustrates the flora that we have associated with us. We've all got microorganisms inside us, about us, and on us. Why do we have them? Well, those in the gut do important things. They create vitamin K for us, for instance. We have organisms that stimulate our immune system. And most important of all, our commensals stop other organisms from colonizing us. The only problem with our commensals is when they end up in the wrong place. And I'm going to show you a few examples of commensals in the wrong place a little later on. Just a few fun facts. We have 50 trillion human cells. But we also have 500 trillion bacterial cells. So we're 90% bacteria. About 180 different species colonize us. And if you are feeling a bit overweight today, 1 kilogram of bacteria sit in your gut. So we do have a special relationship with our microorganisms. I have a special relationship with microorganisms as well. It's a rather more complex one. I'm really interested in pathogens, organisms that can cause us harm. A proteus, avian flu, a prion, some fungi, variety of organisms there. These pathogens can have really serious consequences for us. And some of the consequences are just illustrated in this medley of pictures, and I'm going to talk about some of those. It's really interesting to understand how diverse my field is. When this book, which I'm a proud editor, was first produced by a colleague, Denver Russell and Barry Hugo, there were just 295 pages in that book. On this, the 8th edition, there are 570 pages, telling us that there's been a considerable change in microbiology and that which affects pharmacy. So I'm going to take you on a journey which will show you what fascinates me about microbiology. I'm going to use examples from my own research and some from some amazing collaborators that I've worked with. And what I want to do also is take you on some perhaps tangential journeys, because I've worked with many disciplines, and that doesn't always mean that I follow a straight path in what I do. So on occasions, I'm going to slip out into a tangential journey. So let's start with something which is truly pharmaceutical, and it appeared in the first edition of Hugo and Russell's Pharmaceutical Microbiology. Contamination. I think it's a somewhat reassuring fact to know that probably about 1/4 of the cosmetics that you use are contaminated with microorganisms. The reassuring bit is that they're your microorganisms in most instances. And that's about misuse, quality of preservation. But it's a truism that pharmaceuticals have also been contaminated in the past. And that, of course, is about quality of production. This little illustration here I've got, this bottle here, I think it's a shampoo. It's contracted. That's because the microorganisms that are contaminating it have used up all the oxygen in that container, and its sides have collapsed. Down here, an emulsion, oil and water, normally very stable. Some lipolytic bacteria, those that like to consume oil, sitting at the surface, changing the pH. That no longer suspends. And because these are beautiful images, I thought I'd show you these as well. Prednisolone tablets. You can see the fungi there, in particular in the score marks. Why the score marks? That's where moisture gathers. You add moisture to the substrate of the tablets, you get growth. Those fungi metabolize that prednisolone to an inactive compound. Aren't they beautiful? Look at those fruiting bodies. Absolutely smashing. Here's a bar of soap. Name deliberately excluded, but that's been stored in a moist environment. And here, creams. When you put a cream into a container, you put the top on. When you take the top off, exchange of air, put the top on again. As temperatures fluctuate, you get evaporation from the cream. It distills on the bottom side of the top, and it takes down bacteria or molds from that liner back into the cream. And the water that drips back dilutes the preservative. And there you have a nice nutrient base. So when you have a look at your pharmaceuticals or your cosmetics, just treat them well. And just to give you a sense of the relevance to this clinically, talcum powder, found to be contaminated with Clostridium tetani when newborn infants died because the talcum was used on the umbilicus to dry the wound. Chlorhexidine antiseptic solution, topped up in a pharmacy, becomes contaminated with a very resistant organism, Pseudomonas or Burkholderia cepacia. Burns patients had their surrounding skin disinfected. Nasty organism for burns. I'll come back to IV fluids in just a minute. Antiseptic mouthwash here. Pseudomonas and coliforms, bacteria from our gut. 3 patients on cancer chemotherapy died when they used that mouthwash because they were immune compromised, and those microorganisms were sufficiently challenging for them to die. Now I come to the first of those words in my title, from bags to bones with towers in between. I want to talk a little bit about intravenous infusion fluids. Intravenous infusion fluids, these are breaching our main barrier of defense. You insert a needle through the skin. And intravenous systems can be complicated, depending upon what function they're performing. A route for drugs, fluid imbalance, nutrition. And in 1981, it seems a while back now, in my salad days, I did some work at the Queen's Medical Centre in Nottingham to look at the risks associated with the manipulation and use of intravenous infusion fluids. And we took bags of fluids at the end of their use, when there was still a residual volume in there, and we passed it through filters. And we collected the organisms, placed them onto nutrient agar plate, and let the microorganisms grow on it. Well, it was really interesting. What we discovered here was that in this sort of complex system, a primary bag, a secondary bag, injections being done down here, the risk of ending up with an infusion which also infused microorganisms into you was almost 20%. A serious development of training brought that right down to the national average of 2.5%. The organisms we found were from our hands, from the air, and one patient who was particularly well loved had a bag full of plant pathogens because they were aerosolized in the air and caught up in the manipulation. What we also discovered is this: a cannula taken from a patient, rolled across an agar plate, distributed microorganisms. And this was my first introduction to biofilms. Biofilms are a natural form of living for bacteria and many microorganisms. They do not live generally as free-floating singular organisms. They form consortia. And here is an example of a consortium forming on a cannula from a patient. What I have here is an illustration of how biofilms emerge. Looking at the bottom here, they start with single or odd organisms which colonize a surface. They start to grow. They produce a slime layer. They then consolidate into a larger group. They then form a morphological entity. And then, as they mature, they release fresh organisms, young, vibrant organisms, which swarm away and start the process again on a new surface. And here's an electron micrograph of some probably Escherichia coli that are stuck on the surface, or Pseudomonas aeruginosa. Why do they do it? They can capture nutrient this way, they can protect themselves, they can resist antibiotic therapy, for instance. It's why it's so difficult to eradicate organisms in a biofilm in a person. And they get big as well. Here's a biofilm from a swimming pool, the filter. Here's something that was hanging from a paper processing plant. Yeah, not small. And here, Pseudomonas aeruginosa from a cystic fibrotic patient. So next time you pop into a swimming pool or a hot tub, just feel the sides. If they're slippery, you've got company. Now you might recognize that lad there on the right-hand side. This is one of my tangents. Gordon Stewart, Tony Smith, Gordon Stewart from Nottingham, really close colleague, sadly deceased. Tony Smith from Amersham, and myself. We were part of several groups who took the genes from a photobacterium. A photobacterium, a marine organism, that lives in the eyes of deep-sea fish. And you might have seen them on the film, on pictures. Eyes open, glows, eyes shut, dark. It's thought this is a way of catching prey unawares. A fish will swim in front of a prey, open its eyes, it's seen, then nip around the back with its eyes closed. Apparently its eyes closed. They're not, of course. They've just shut this little shroud which conceals the photobacterium living in a symbiotic relationship. You can take the genes out of those and you can put them into bacteria, into terrestrial bacteria. And this is what we were doing. And they make those bacteria glow. As they grow, they glow. And what's really interesting is you can produce a plate of growing bacteria, and instead of it just looking like a cloudy layer of agar, here you have them glowing, and here, where we've put antibiotics in place, you can see it's killed the organisms. Dead, no glow. We were doing this work at around about the time that Legionnaires became a really significant issue. First in the States, I think it was in the 70s in Philadelphia, first discovery. But subsequently, any air conditioning system which produced a wet surface of a slightly elevated temperature was susceptible to Legionella growth and Legionella biofilm. You might remember back to one of my earliest images there of a hand with some slime on it. That was drawn out of a cooling tower pit. A problem with all of these towers and air conditioning units that have exposed water is that they're susceptible to Legionella. They have to be controlled by biocides, antimicrobial agents, to stop those organisms from growing. And it's failures in biocide control which led to aerosolization of Legionella from biofilms and the subsequent problems that occurred. And the trouble with Legionella is that at those days it was very difficult to grow, and it would take 5 days or so before you knew whether you got a contaminated area. And so it would take a while before you knew whether your biocide control was working. So with Amersham, Gordon and I worked to try to produce a rapid biocide test system. I do apologize for this slide. It's the only one in existence, and it's a 35mm slide, which I've had to have scanned. But we used a series of bioluminescent microorganisms, and we dosed them with different levels from a cooling tower, water from a cooling tower. And we looked to see when the light went out. 30 minutes it took, not 5 days to grow the Legionella. And through a series of days here, a bit difficult to see, but all of these tall peaks here represent viable counts, 5 days late. And this represents bioluminescence in our test system 30 minutes after taking the sample. And over a series of days, you can see that by adding more biocide, constantly adding more biocide, you eventually brought everything under control. But we could tell you that very, very rapidly. And Amersham did a lot of work on this in cooling towers. And so now, back off the tangent and back to a theme of biofilms and medical devices. At some stage in our life, we are likely to be invaded by at least one of these medical devices. Sutures, catheters, contact lenses, prostheses. All of them provide a new opportunity for microorganisms to colonize and infect. Why are they so risky? Well, of course, all of those devices are indwelling. They breach our normal host defense of our skin. All the barriers are bypassed. They provide a very unusual environment that bacteria don't normally meet in a person. If you've got an artificial valve, you are at high risk of a streptococcal biofilm forming on that valve. That's because when you brush your teeth, You put 100 to 1,000 bacteria into your blood circulation straight away. They're mopped up, they die quickly because they're from your own flora and the immune system is very effective. But if they can get through to a foreign body and attach to it and form a biofilm, they can stay. Because they produce slime and antibiotics don't solve it, you can't get your immune system to combat it then. And very frequently the materials that we use in these systems have been designed for mechanical reasons, but not to avoid microbial attachment. Now, I've been fortunate enough to work with some surgeons who were looking after patients with kidney failure. And one of the ways of treating, often temporarily before kidney replacement, is to use continuous ambulatory peritoneal dialysis. And for me, it was an exceptionally useful model for understanding microbial pathogenicity. So here we have patients undergoing CAPD. And what you've got is you've ostensibly got a peritoneum here, which is an open space surrounded by a membrane in your abdomen, which is well fed by capillaries. If you fill that with 3 litres of a dialysis fluid, the waste products and the toxins that are circulating in your blood permeate across that peritoneal membrane into the dialysis fluid. And so for about 6 hours, you have this 3 litres inside your peritoneum. And after 4 to 6 hours, you drain it out. So you've got an indwelling catheter, which goes into the peritoneum. You then drain it out. You put a fresh bag on, and you do it again. And then at night time, instead of a 4 to 6 hour change, it stays there for 8 hours. Amazing. Keeps people ambulatory, keeps them well. But there are consequences. Here's the tip of a catheter taken out of a patient who was suffering a peritoneal infection. And you can see, look at this elegant biofilm here, 150 microns thick. It's beautiful. Staphylococci come from the patient. In studies that we did in Nottingham and in many other places, and in Brighton, this is the sort of flora that you can get from peritonitis. About half of these are staphylococci, organisms that are associated with us. Everyone who is undergoing CAPD is likely to have an infection at some stage. The rate of infection used to be twice a year. It's now much better. It's probably 1 every 12 months, 18 months, or even better. But for some patients, they don't get that sort of relief. They get frequent infections. Staphylococci cause a lot of pain. Fluid becomes opaque, discomforting. Pseudomonas aeruginosa can be life-threatening. But that peritoneal membrane, every time you have an infection, is made just a little less permeable. It's a little less functional. So you lose the capacity eventually for CAPD. One really great thing about this is that you can get used dialysate from patients by the bucket. And of course you can buy fresh dialysate from the suppliers. So that gives us a chance of modelling the peritoneum to see how bacteria behave. This is your peritoneum in model. So there's your peritoneum. There's some fresh fluid. This is used fluid. And this is a receptacle to gather waste. And what we did, I won't go into the real detail of it, but when you add fresh fluid over a 6-hour period, it changes. It changes its composition because of all the stuff that's dialysed into it. So we tried to follow that, to try to model that change by adding fresh fluid in and then beginning to titrate in used dialysate from patients. And any excess, this is where the fluid gathers, was just run off to waste. But we could put catheters in here, we could take samples from in here, and so we could follow the bacteria as they grew through a process of peritoneal dialysis. And of course, we could follow just what happens in real life. We could then, after 6 hours, put in a fresh bag of fluid, run out all the other stuff as we displaced it with fresh fluid, and then go through the cycle again. A complicated slide, and you don't need to know. I'm not going to spend time going through every part of this, but I'm going to tell you two things. What we discovered is that those bacteria that adapted to the life in that model, same as adapting to the life in our peritoneum, elaborated a sophisticated mechanism for capturing iron. Iron is one of the most plentiful elements in our body, but one of the most inaccessible elements in our body, because it's captured in hemoglobin and other chelating agents. And for a bacterium that needs iron to live and to grow, it has to steal that. And so it creates, if it's going to survive as a pathogen, a more powerful chelating agent, iron-gathering agent, than hemoglobin. And here it is. We can detect it by a particular color on a particular type of agar. 3 staphylococci put into our model. They don't, in nutrient broth, which is a normal conventional laboratory medium, create those, but they do in our model. And they do when you take them out of the body. And they've made the agent which goes out and gathers iron, and on their surface, they've grown 2 new proteins which capture that chelating agent and bring it back inside them, and then they break the chelator down and release the iron, and they grow. That's the first message. These microorganisms adapted to live in an environment which was iron-constrained. The second is that as we put fresh fluid in, and that changed, any microorganisms that had got in there by accident started to grow. But they were quite hydrophilic at that stage, water-loving, quite happy to float around. And so they dispersed all around the peritoneum and around the cannula that was in there. And then, after they'd grown to maturity, this is particularly the case for the long overnight growth, they'd become hydrophobic and they'd stick. And then you put your new fluid in, flush out all the old fluid, but the bacteria stay. They're not removed. And so what we had is a microbial mechanism in order to retain and survive in this environment. And once you put new fluid in, they became hydrophilic again, and they rushed off to all the other corners they hadn't colonized. And so the cycle happened again. Amazing. So with models like this, what should we do? We want to try to control microbial adhesion. You can't control it to the peritoneum, but you can control it to the catheter, which is often the major location for infection. And so some of the work I'm going to talk about now was done at Brighton with some excellent colleagues. And just to prove, this is Cockcroft Building, to prove I was here, there's my Range Rover, CR43 URS, or sorry, C43 URS. So I was here at the time. And what did we do? Well, we investigated how bacteria stick. They stick by charge, they can be repelled by charge, but they can stick through charge. They can stick together because it's a hydrophobic environment and they love sticking and eliminating water and sticking together, or there can be a very particular specific attachments to a surface. And read for this surface, a cannula. So we're able to modify these surfaces in different ways, and just a couple of examples of how we've done it. This graph looks at adherence for a range of species of bacteria, and these tall histograms here are attachment to surfaces unmodified by chemical gas and oxygen gas, and here modified surface. And we took a surface of polystyrene, we occluded one area, this area, and treated this area here with an oxygen gas to change its chemical character, but left this one untreated. Flooded it with bacteria, stained the bacteria, and you can see the definite difference there. We've also taken surfaces and attached to them Pluronic block copolymers. That's a polyethylene oxide, polypropylene oxide, polyethylene oxide compound. And that orientates at surfaces with the polypropylene oxide hydrophobic part on the catheter or substrate surface with these hydrophilic chains sticking up. A bacteria can't attach. Steric hindrance, it's pushed away. And then finally, and this is a slide courtesy of Andrew Lloyd, we can try and confuse microorganisms by taking a tip from red blood cells. And this is work that was done with a company called Biocompatibles. Take phosphorylcholine, which is a component of red blood cells which don't get colonized by bacteria, and put it onto the surface of different materials. And here's a material coated with phosphorylcholine, very few microorganisms, and here's one that isn't. Biomimesis, great fun. And then finally, in terms of antimicrobial control, some research which we're currently doing, a research student, Jack Barker, with colleagues Ian and Cressida. We're looking at it. It's work that Jeff Hanlon did for many years at Brighton, and I was involved in as well, looking at the use of bacterial viruses to control contamination. Here's an example of a bacterium. Here's an example of its virus called a bacteriophage. Bacteriophage comes along, sticks to the bacteria, shoves in its DNA. It is then uses the synthetic machinery of the microorganism to make new parts to it. These, when assembled, destroy the bacteria from inside, and they're all passed out. And then they are lytic, they kill other bacteria. We want to attach them to surfaces and see if that can actually decolonize surfaces. And here's how we do it. Here's a surface. Here's a charged polymer, which sits on the negatively charged surface. It also attaches to on the negative head of a bacteriophage and orientates it in just the right way to inject the DNA into the bacterial cell. And here's an example. There's a disc here on an agar plate which is covered with bacteria. This disc has been flooded with bacteriophage and then rinsed, but it has no coating on it. And you get just a few zones of clearing. This is some residual phage which is released into the plate and destroys the bacteria, producing clearing. Here is one where we've put on our charged coating, the bacteriophage sit, and every time they meet a microorganism, they infect it. So the plate is virtually cleared. And a final brief tangent before I come into the concluding track and discover bones. Surgical instruments, we're all the beneficiaries of surgical instruments. And surgical instruments can end up becoming contaminated with biological material, sometimes infective biological material. Many of you will know about bovine spongiform encephalopathy. Farmers feeding sheep that had scrapie to cattle. And that scrapie prion, a bit like a protein virus, entering the cattle, causing encephalopathy and some dreadful examples of cattle dying. Well, in 1996, the first variant Creutzfeldt-Jakob disease was recognized. And that is a disease where the prions, which normally never get from sheep to us, used a vector, the cow, and entered us, and caused devastating illness in very young adults, average age about 28. The clinical cases are now being gradually declining because all of this practice has been halted, but carriers still remain. And the key thing is, we think now infection will pass on through contaminated medical and dental instruments. And this is how a prion works. We have prions in our body, natural prions. You get a foreign disease-causing prion. It comes close to one of our prions, and rather like a string of dominoes, it changes the shape of all the good prions into the shape of the bad ones. And those cause real morphological damage in the brain and injury. Here's some medical devices, some scissors. The government established a committee with a snappy title, Engineering and Science Advisory Committee on the Decontamination of Surgical Instruments, including prion removal. I was a member of that committee, and I was there to look at the sterilization and disinfection that we might use to protect ourselves. And what we eventually ended up with is recognition that prions were the most resistant organisms to the heat sterilization process that's used for bacteria we've ever seen. They are 1 trillion times more resistant than the most resistant bacterium. And the only way we could reduce the risk was to stipulate that surgical equipment had to be rinsed, go through an automated washer disinfector, moist heat sterilization process, and that produced an overall reduction of up to 9 logarithms. So if you started with 1 million prions on a piece of medical device, you'd end up at the end with a risk of 1 prion remaining in every 1,000 pieces of equipment. And so we added to this by saying single-use instruments whenever possible, destroy all neurological and retinal surgical instruments, and make sure that is followed. And by 2017, the total number of recorded cases of VCJD, variant CJD, was 178, and they're all now deceased. The only mode of transmission now is through surgical equipment. 1 in 2,000 of us carry infective VCJD prions in latent form. So now, to the end of the journey, bones. Been very interested in hip and knee transplants. Not transplants, replacements, sorry. 1/3 to 1/2 of total knee replacements use cements to keep them in place in the bone. And that cement comes in a packet. It has an oily liquid called methyl methacrylate. It has a powder, polymethyl methacrylate granules, and it has a catalyst to make the cement. And you mix it up. Any of those who have old cars will know about plastic padding. It's exactly the same as plastic padding. And so you mix it up in the surgery, then you pop it in there in order to locate the bone. And one of the risks is bacterial infection. So, manufacturers of bone cements have said this will be a brilliant vehicle for putting antibiotics in. But antibiotics are water soluble. They can't put them in the oily methyl methacrylate. So they have to grind them up and put them in the powder here. And dispersing that is a very, very inefficient process. So working at Cardiff, we developed a system where we put phospholipids together to form a vesicle called liposomes, well known in the drug delivery world. But those liposomes are hydrophilic on the outside, hydrophilic on the inside, where you can put a water-based drug, but they're hydrophilic on the outside, and they will not disperse in oily methyl methacrylate. But if we add those branched copolymers that I talked about, polyethylene oxide, polypropylene oxide, polyethylene oxide, they attach to the outside of that liposome and produce a hydrophobic area on the outside. And so then you can mix it in the oil, you can mix the oil with the powder, and you get beautiful liposomes throughout the powder. Here is an example of a liposome with the antibiotic in it. And this is what the conventional powder looks like, great big granules of gentamicin. That's the level of gentamicin released over a number of days from conventional products. That's the level it's released from our own. And just to demonstrate, these are some zones of inhibition around little pellets that we've made with antimicrobials included in the liposomes. This is a conventional bone cement, and you can see you get big zones, no zones, and almost and very little zones. That's because of the dispersion of the antibiotic in there. And work that Matteo Santin has been doing in this institution has been looking at some of the phosphatidylserines, which are a type of phospholipid, and they can generate mineralization and prospective bone growth on titanium. Another potential that sits here. And now we're doing some work in Cardiff looking at dental cavity treatment, composites containing these liposomes for both animals and humans. And now I have to acknowledge two things. I have to acknowledge that I've probably kept you here for 8 minutes longer than I should have done, so I apologize for that. And to thank you very much for listening. I must acknowledge that I have been so fortunate with the colleagues that I've worked with. Incredibly fortunate. I've listed some of my close collaborators there who've made every minute of research challenging, exciting, varied, and worthwhile. And an incredible array of postgraduate students and postdoctoral students who have really been the engine room to the work that we do. So thank you very much for listening. It's been great talking to you.